Mr. Vice Chancellor, or the Deputy Vice Chancellor, the Registrar, and all the deans and heads of departments here present. I am honored and pleased that all of you are here with me today in person. To listen to my talk as part of your Faculty of Science guest lecture series. I am humbled and honored, and I thank that's the Vice Chancellor, who is the chief host. I thank also the Dean of Science, and I thank my good friend Professor Woods, who was involved in us this meeting today. I am aware that the major this mission of this great University of Ibadan is to foster education through these various means. This including
this is better.
Okay. All right, I will continue now. Ladies and gentlemen, I would have loved to talk more about that's guest lectures in universities, and to compare the guest lectures here at the University of Ibadan with other universities. However, I do not have the time to do so today. So I would proceed to the lecture that we have in front of us. In preparing this lecture, I took into serious considerations of the diverse backgrounds of the audience. However, to understand this lecture fully, one needs to understand the rudiments of medicinal chemists, oncology, drugs, and the language thereof. However, I decided to emulate Einstein's dictum, which states we should make things as simple as possible, but not simpler. So therefore, I will now proceed to today's lecture. So you can see my title out there, The Discovery and Development of a Novel Molecule. That's Galeterone and the Next Generation Compounds. In it also, I have, some subtitles, which is ingenuity or serendipity, or is it the other way around? And I will also discuss a little bit about academic, that's drug discovery and development. My talk will be divided into two parts. The first part will discuss, that's Galeterone, from the bench to the bedside and its resurrection. the rational development. I'll just give a summary of the key preliminary data, a summary of the clinical trials, and its current status. In the second part, I will discuss the next generation Galeterone analogs. Again, I'll provide, there's a rationale for the next generation compounds, summary of the key data and development of, the Galeterone salts. I have to make this disclosure, which is based on the requirements, from my university. Okay, that VN124, also called TOC001 Galeterone, was invented or discovered at the University of Maryland School of Medicine in Baltimore. And it was discovered in my laboratory with me as the lead investigator. And I have a co-investigator, although she's late, Angela Brody, shown here in this photograph. This technology was licensed by the University of Maryland, Baltimore. That's to Tokai Pharmaceuticals in Cambridge, Massachusetts. There's now, a new licensing to a company called ESL, but it's now transformed to LTN, this pharmaceutical. And this, here was the signing ceremony. The university president is signing this agreement, and the heads of Office of Research and Development, Jim Hughes. This is the CEO, Dr. Lowe of LTN, and that's the CEO of the company. These are required for me to give any public speech. As the title states, the development from academia, academic drug research. Some drug discovery research and efforts are anything but academic. If you define the word research, means a careful or this diligent search, a studious inquiry or examination, very nice and noble words. Academic is a horse of a different color. In its noun form, it means a person who is very learned but inexperienced in practical matters. Adjectival form means having, no practical or useful significance. Synonymous, with the word pedantic. Typically, when you say something is academic, you mean it does not matter. So it means that, Mr. Vice Chancellor, so from the dictionary word of academic, it means that we are all here in an academic institution and all we are doing, does not matter. Is that really true? No, of course. So I tell myself, I want to do things that matter. I certainly don't want to be associated with a term like pedantic. And as you would see, in the next discussions of the research that I have done in academia, you will really say that that dictionary definition of the word academic should be revised. In my laboratory, this is the overall research strategy. We have the medicinal chemistry laboratory, which is involved in target identification, rational design of molecules. We're able to synthesize our molecules, and molecular modeling. In our pharmacology oncology lab, we're able to test our molecules first in vitro and in animal models. And the training, of course, that students get will be in organic and medicinal chemistry, in onco-pharmacology, experimental therapeutics and cancer biology. The goal, of course, is to move these molecules into clinical trials, and we're hoping that we'll get these new drugs. This typically, for the preclinical studies that we do, you have to start out with an hypothesis. Say, for example, a compound A will latch onto a protein B and disrupt this tumor growth. That's the hypothesis. Of course, you cannot develop anything without funding. You have to write grants to get funding. Then we carry out, the in vitro studies, in models, appropriate models of cancer. And if we get the lead compound from this, we move it to test in animals. And of course, the goal is to go into clinical trials. The phase 1, you test for safety, phase 2, efficacy, and in phase 3, that is a large-scale trial, and if it passes phase 3, then your molecule gets approved by the Food and Drug Administration as a drug for human use. I'll just summarize the way we started out with the discovery of Galeterone. We wanted to identify a specific inhibitor of an enzyme, CYP17, for prostate cancer therapy. Androgens will make, that's prostate cancer to grow. And the idea is that if you inhibit the CYP17 enzyme, you will reduce androgens. That's to castrate levels and tumor dysregulation. So what is known is that, call this a prostate cancer tumor. The major sources of androgens from, that's the testis, that's 90%. From the adrenal glands, about 8%. And we now found out recently that the cancer cells themselves are also able to produce androgens. This here shows the male reproductive tract. And the prostate gland is shown right here. It surrounds, that's the urethra. It sits, that's below the bladder. So Charles Higgins won the Nobel Prize in Physiology and Medicine in 1996, for his discovery, that castration of patients will reduce their tumors. So what he discovered was that if you remove, the testes, you reduce, of course, the levels of androgens. That's from this source. But remember, I told you, you also have these other sources of androgens from the adrenals and the cancer itself. As an alternative to castration, we then began to seek to develop inhibitors of CYP17. And the idea is that with inhibitors of CYP17, you will be able to block the production of androgens from all sources that are known. This are the different stages of the disease. It starts out as a small tumor, and then it would start enlarging and it spreads. What kills the patients is when the cancer, has now metastasized to other organs like the liver, the bones, and all that. And if you look at, these statistics, the 5-year survival rate of U.S. men, metastatic prostate cancer is 28%, compared with almost 100% with it is confined. So the idea of trying to develop molecules that would inhibit, that's metastatic disease, is an important goal. So what happens when you inhibit the CYP17 enzyme? So in this slide here, we're starting out, that's with cholesterol. Say, the CYP17 enzyme is shown here in 2 steps. So what happens when you inhibit this enzyme system? So when you inhibit that, there's a downregulation of androgen and testosterone. I must point out, remember, sometimes you hear people tell you, okay, that cholesterol is a bad thing. If you don't have this cholesterol in your system, you will not be able to produce what we refer to as the male sex hormone and also the female sex hormones. So, okay, that's cholesterol, of course, is not a bad thing. It's required for the male and female sex hormones, the synthesis. These androgens would have to bind, to a receptor, the androgen receptor. That is derived from chromosome X, which means you can get it from males and females. That's the full length, and you have the splice variant. During the last period of this COVID-19, you heard of, okay, that's the splice variants. So in androgens also you have the splice variants, and they're important in resistance of the disease. So what happens is that the androgens would bind to the androgen receptor. There's a phosphorylation, there's a dimerization, and the binding to the DNA response element. And this, of course, will stimulate the growth of the disease. So what we are doing is to inhibit these actions. And I must remind you that in prostate cancer therapy, the mainstay, the current mainstay of disease, of treatment of the disease, is a blockage of the action of the androgens, to the prostate. That's the currently approved drugs. So the synthesis, you have to design, how to make these molecules. And that was based on our knowledge of the CYP17 enzyme, which has an active site and a heme prosthetic group. We are also familiar with the reaction that takes place. From pregnenolone, for example, you are cleaving this side chain. It involves these two steps. So what we were doing was to design inhibitors which would block that action. Okay, the crystal structure of this enzyme was not known at the time when we're doing this design, okay, but it's now known. So what we decided to do was, in this region of the molecule of pregnenolone, this region, that's where the cleavage takes place, or the action. We now wanted, okay, that's to modify this region to develop inhibitors of the enzyme. So with groupings, we can form a coordinate bond to the heme ion so that you inhibit that enzyme system. That was our design strategy. However, when you design these molecules, you have to synthesize the molecules, and that's through chemistry. We made this discovery of a novel reaction, this nucleophilic, this vinylic addition, elimination, substitution reaction, of this substrate. So I'll just explain to you what happened because it's important. So we had this molecule and an acetate group. And the experiment we're doing here, was just to hydrolyze this hydroxyl group. And the idea was to get this molecule. But notice what we got now was compound 8, in which the Cl has been displaced, and we have, that's a methoxic grouping at C17. So based on this idea, we figured out that if we can have azoles in the system, we will be able to make these novel compounds. At the time we synthesized this first lead compound, this was the first molecules, these steroidal molecules, in which an azole group is linked to the C17, with a nitrogen atom. This prior, they were linked, via a carbon atom. And with that, we were able to discover our lead compound. That's Galeterone. So from my subtitle, ingenuity and serendipity, or is it the other way around? This slide, here summarizing the synthesis of Galeterone from a known compound that is sold, that's commercially. This synthesis is shown here, which I'm not going to go through significantly. But the important thing is that this synthesis, it, it's what is currently used, okay, that's commercially, to make galaterone. So the impact of our molecules was shown in different, these models, of prostate cancer, where we compared our molecule, with castration and some known drugs. And the summary of all these in vivo studies, which were independent, shows that, okay, that galaterone is better than some blockbuster drugs currently used. And of course, in this slide, here just summarizes what we found out. We're planning to develop CYP17 inhibitors. We also found out that this, would antagonize the androgen receptor. They would also induce the degradation of the androgen receptor. And in further studies, we also found out that they would impact on some pathways which do not involve the androgen receptor. And based on our hypothesis, we now found out that they would inhibit, that's the protein translational complex, which I will discuss in the next few slides. Again, all we did was to follow our scientific data, and we didn't just close our eyes to our original hypothesis. The question again, ingenuity and serendipity, or is it the other way around? So in the next few slides, I will discuss, that's the clinical trials of our lead compound, Galeterone, that was licensed, that's a Tokai pharmaceutical to develop. So this slide here is a phase 1 study in prostate cancer patients. And the PSA is a marker, that's for prostate cancer, this prostate-specific antigen. And you can, identify this, from the blood of patients. So this plot is called the PSA, waterfall plot. So if you take this line, that will be the starting point. The bars you see here are patients, that responded to the therapy, and there's a decline of their PSA. These are patients that did not respond. So this, was very promising. And, Galeterone earned, what you call a fast-track designation by the Food and Drug Administration of the FDA, which means that the trials should move forward, and the phase 2, this trial started in 2012. However, the requirement for this PSA reduction required quite a high dose of the compound. The next few slides would show the impact in a phase 2 study, of Galeterone, in patients who were, some are resistant to current drugs, and so on and so forth. This trial was called this ARMOR2, androgen receptor, this modulator optimized for response. These are the different classes of the 4 patients. The naive patients, and these were already refractory to FDA-approved drugs. And the monitoring, again, is a PSA level. This is a waterfall plot, and you can see that this many patients, did respond to the therapy in phase 2. While we were developing Galeterone, there was an approval of a drug that inhibits the CYP17. That's abiraterone, this Zytiga, and this was the dose used. There was also another molecule, it's a blockbuster drug, enzalutamide or Stanzia, which blocks the action of androgens at the receptor. However, these drugs are ineffective in patients that express the AR splice variants. Remember I mentioned that in resistance in prostate cancer patients you have the splice variants, but from the summary I showed to you, that Galeterone is effective in prostate cancer that does express the splice variant of the androgen receptor. So this again is the figure of the androgen receptor, and you can see that both the full-length androgen and the splice variants are degraded, by Galeterone. So we did additional studies, and this work was published in Journal of Medicinal Chemistry. Our article, made the cover page, actually, where we are showing that our molecules are able to degrade the androgen receptor and the splice variant. The Journal of Medicinal Chemistry, is the most cited medicinal chemistry journal in the world. So based on the results that we showed in the lab and also the phase 1 and 2 clinical trials, Tokai Pharmaceutical went and did a study of their phase 2 data and identified, there are 7 patients that had the splice variant androgen. And of the 7 patients, 6 patients, responded to the therapy. So in their phase 3 clinical trials, they now wanted to focus, on just the patients that expressed, as the splice variant, which was against, my advice at the time. This clinical trial, did not succeed, although, the drug, was safe in the patients. So in the next slides, I will discuss why Galeterone did not succeed in phase 3 clinical trials of prostate cancer and what is being done to this rescue. There are complaints about the design of the phase 3 clinical trials. The biggest, was the sample size. So the other 2 drugs I just showed to you, which were approved around when we were developing Galeterone, the number of patients in their phase 3 was very high, over, that's 1,000 patients. But Tokai, in their phase 3, just had very few patients in the trial. And another issue also was the top people in the company, when they were designing phase 2, were selling their stocks from the company. However, based on the data that we have, we believe that the failure in phase 3 by Tokai, was because of the trial, this design. So this slide, just summarizes what we know about, that's Galeterone, the failure in phase 3. And the recommendation to rescue is that we should conduct another head-to-head phase 3 study, which is a large-scale study involving, this many more patients. And that's where, so on this slide, of course, I'll give credit to my graduate students and former postdoctoral fellows who worked on this project, which are shown here, all are frustrated. So plans, to rescue this Galeterone is shown here, where we were able, to secure, that's tablets from Tokai. We needed funding for the storage. That was provided by David Weber, who is the director of our CBD. And this, again, I already showed you, where it's now licensed to this LTN, with pharmaceuticals. So LTN is planning now to conduct some new clinical trials. The PI of the study is Arif Hussein, who is in our cancer center. He's an oncologist in our cancer center. And the plan is that the new phase 3 would begin soon in Jamaica. You would ask the question, why Jamaica? It turns out the CEO of LTN is from Jamaica, and you may be surprised to note that the Caribbean has the highest death rates, actually, from prostate cancer, shown here. So that's the flyer for LTN for their phase 3. That's the clinical trial endpoints and secondary endpoints. And as I say, these trials would commence in Jamaica, and there are plans to conduct trials in Nigeria, in South Africa, in Tanzania, and in Uganda. LTN also has the rights to develop the Galeterone salts, which I will discuss a little bit later, just show you some new data. So, in the second part of my talk now, I will discuss the next generation Galeterone analogs. So you would ask the question, what is the rationale for developing the next generation Galeterone analogs? When you develop a drug, you do not just sit on your house. You ask yourself the question, can we improve on this lead molecule, which is yet to become a drug? And if we consider that the dose used for Galeterone in patients was quite high, so that was a stimulus to start looking out for the next generation, these compounds. And the idea is to increase their anti-cancer potency, improve the pharmaceutical, these properties. So this was a paper we published in JMed Chem again. The take-home message here is that when we did modifications around the benzimidazole grouping, around ring D, and also ring A and the side chain, we found out that you can increase the side chain here where you have R and you still can enhance, that's the potency of your molecules. And we developed some lead compound, the VNPP414 and the 433, which is already published. But the problem here was that this discovery synthetic route, the yields were very low, 12 and 11%. So the solution, we were able to improve on the synthesis of these molecules. Remember, this was the discovery route, 11 and 12%, but we have now been able to increase it this sixfold, for both lead compounds. This slide just shows you the synthesis, which I'm not going to spend time on for the sake of time. We already have applied, that's for patents to protect this methodology. And this slide here, this summarizes the mechanism of actions that we already know with Galeterone. The androgen receptor axis and the androgen receptor independent axis, where it does impact on cyclin D1, MNK1 and 2, EIF4E, is translational, is complex. Okay, so this slide is shown here about the MNK. For the biologists and cancer biologists, the RAS-MEK-EC pathway, PI3-AKT pathway, is upregulated in most cancers. So what we found out is that Galeterone and its analogs are able to impact also, on protein translational, this complex. And if you inhibit the protein translational complex, the implication is that this pathway has also been shown to be upregulated, that's in prostate cancer. So you can see this here is resistant prostate cancer, and the EIF4E is upregulated, shown here by the Western blotting, and it's also shown in these cancer patients. And in the clinic also, it shows that the patients that express the lower level of EIF4E have a better, that's prognosis as compared to patients that have a high level. This is survival now, and this is in the period of in days. So it makes sense also to distarget that protein translation. However, in targeting this protein translation, you may want to ask the question, why won't inhibition of EIF4E kill all cells and not just the cancer cells? Not just the cancer cells. The reason is because there are two major classes of these RNAs, mRNAs. During the COVID, you heard a lot about mRNAs. They are the strong mRNAs, like the housekeeping genes, and they are the weak mRNAs which are involved in diseases and in cancer, like VEGF, cyclin D1. The strong RNAs are well translated, even when the availability of EIF4 is very limiting, while the weak mRNAs are translated when EIF4A is increased. And if you reduce the expression of EIF4 in tumors, you inhibit cell proliferation, you suppress tumor angiogenesis, you repress the movement of cancer cells, Yes, so you repress metastasis and induce cell death. So when you reduce EIF4, you have minimal effects on this total protein and therefore minimal effect on normal cells. So this is another way of explaining the inhibition of mRNAs. During normal growth, the EIF4A is very limiting. You can see that the normal cell, the strong mRNAs are fully, that's translated. However, in tumorogenesis and in metastasis, you need an increase, these levels of EIF4A. So when you inhibit EIF4A action, you are just reducing the cell proliferation, angiogenesis, metastasis, and increasing apoptosis, that's cell death. So again, this is what I already talked about. And we wrote a review article which shows that you can target the protein translational complex as a new paradigm for treating breast cancer. The ongoing studies which are being developed for the next generation are supported by a grant from the NIH, and we also have some additional funding also from the NIH. So in the next few slides, I'll just give you a summary of the lead compounds, the VNP, the next generation GAL compounds. They inhibit the cell proliferation, this colony formation. Oh boy. You want to take out, that light is causing. Is there a way you can take out the light from there because it's. Okay, I'll continue for the sake of time, right? So you can see, we're able to characterize our molecules which would inhibit both the drug-naive and the drug-resistant cells. That's shown here. That's this colony formation. By Western blots also, we're inhibiting, the molecular markers MNK1 and 2. Okay, thank you. We're able to inhibit the EMT. Remember that in cancer, when cells migrate to other organs, that's what really kills the patient. And as you can see here in these in vitro models, we're able to inhibit, that's the migration. This is the control where there's no treatment. And look at the treated ones. It inhibits the migration and invasion of cells. That's the control. Our molecules are able to inhibit the cell migration. That's the quantification of the cell migration. Then, of course, you want to do a PK studies. That's Galeterone given in various ways, IV, IP, and oral, and that's the next generation compounds. If you want to highlight on 433, we're able to improve on the half-life at 25 times. The oral, you can give it orally, increased by 2 times. Cmax increase, and the area under the curve is also increased. And the next thing, of course, we did an in vivo studies, which shows, and we compared it with an FDA-approved drugs, which is not statistically significant from the control. But our molecules inhibits the cell growth, the next-gen compounds, this dose dependently. We did additional studies, which I'll just move forward, where we overexpress the androgen receptor, and we are still able to inhibit the cells. We showed that it's through the degradation of the MNK. The last section that I want to discuss, which is very important, we are now developing, the hydrochloride salts of Galeterone and the next generation compounds. So you can see that's Galeterone. We have the Galeterone HCl, the 433, the mono HCl, and the di HCl. And as you can see from the slides I would show, we're able to increase the efficacy. An analysis of FDA-approved drugs shows that the 48% of drugs are sold in the salt form. And the HCl, in fact, leads, and that's why we focus on the HCl. And shown also in cancer drugs, the salts take precedence. So this slide, I'll just go through it. These are cell lines here in green, from white patients. These are cell lines, from black patients. In the bottom line here is that we have shown that our molecules are effective against the cell lines that are derived from either the white or the black patients, irrespective of, that's ancestry. So which means that if we're able to develop these molecules, we would not just say that they are effective in the white patients, they will also be effective in Africans. So, this slide just shows the molecular markers of the salts, and if you just eyeball it, they are superior to the parent compounds. The first thing we did when we made the salts, that's a pilot study of our molecules. That's a control where there's no treatment. That's Galeterone at quite a high dose, 100 mg per kilogram twice a day. This is the salts of Galeterone at 22.6 mg once a day. And these are the salts of the next generation, these compounds. So one of the first things you look at when you conduct animal experiments in vivo to test your compounds, you look for the body weight of the mice. If the compounds, are toxic to the mice, there will be reduction in body weight. And as you can see here, there's no difference between these treated groups and the control. So that gives you an indication that these molecules are safe at this effective dose. For Galeterone, we could improve the dose efficacy by 9.7-fold. For the 433, we could increase the dose by 4.3-fold by just making the hydrochloride salts. Then we did some more rigorous studies, but we compared it with approved FDA drugs. That's docetaxel and enzalutamide. These are the tumor volumes. This is the number of days of treatment. These are the tumor weights at the end of the experiment where we take out the tumors and weigh them. And these are also the tumor volumes. You can see that in some of the groups, that's the Galeterone salts. There's regression of the tumors from where we started. And these are the individual tumors from the mice. Again, you can see the body weight. It shows you that these molecules are safe at these effective doses. And again, for the 433 and the salts also, we did the same thing. And as you can see, there is this regression of the tumors. And our molecules, the lead salts, are more effective than FDA-approved. That's prostate cancer drugs, as shown in this slide. So this slide then shows the tumors at the end. These are FDA-approved drugs. They are blockbuster drugs. That's the control. That's Galeterone and the salts, and the VNTP 433 and the salts. Clearly, this shows you that our molecules are more effective than FDA-approved drugs. And the biomarkers also are important because you want to make sure that the molecules are hitting, okay, that's the targets, okay, that you intend. As you can see, these compounds are quite effective and the biomarkers are significantly depleted. All right, so the AR, the androgen receptor, we've been discussing it in terms, of prostate cancer. However, some breast cancers have been shown to also express the androgen receptor, and the androgen receptor is important. Remember that breast cancer, would affect all humans. It's not just females who are afflicted with breast cancer. It also affects men. So on this slide on the right, there are clinical trials ongoing with FDA-approved drugs. Okay, that's for prostate cancer, but they are now testing them. Okay, that's in breast cancer patients. That's the patients where they show that they express, okay, that's the androgen, this receptor. The other groups also just working on this. So I'll just move on to the next slide. So we did a study. This is a breast cancer cell line that expresses the androgen, this receptor. And as you can see from this curve, our molecules are quite effective also against this. And you can look at the tumors. And here also, we compared them with FDA-approved drugs. We compared them with FDA-approved drugs, and our molecules are better. We also have another class of compounds which we're developing, and you can see these compounds also inhibit this breast cancer. I'll skip this slide. These are some recent publications in our lab. This is a summary of the data that I've been talking about all day. So, the take-home message is that we have developed these potent molecules, and the salts are proving to be much better. I think I'll skip this slide too. Okay, usually when you develop molecules, if you want to advance them into clinical trials and develop them as drugs, you have to protect them. So your university should be able to invest funding, protecting, okay, that's the technologies. And you can protect your technology if you apply, okay, that's for patents, both the local and international patents. So that's very important. So based on all the discoveries in my lab, we now decided to partner with the University of Maryland, Baltimore, through the Office of Tech Transfer. We formed a company called this Isoprene Pharmaceuticals, where we're now beginning to advance our technologies. So here you see me described as the president of Isoprene Pharmaceuticals. You might be asking the question that, okay, Vincent NJAR is a professor at the University of Maryland. How come he is a president of a private company? Okay, so what takes care of that is that the university has okay, what you have an approval of this conflict of interest. So there's a management plan for that. So this university, I would advocate, should also have a means, where professors and faculty are able to form, okay, that's private companies, either on their own or in collaboration with the universities, as long as you have a conflict of interest, okay, management plan in place. That takes care of that. So that's what's happening. So I'll just go on. Our company, okay, has a 2-year, okay, that's $2 million, the small business research grant from the NCI. We're trying to develop our first molecule of the company, and we are collaborating with another CRO, okay, that was pharmaceuticals, who is helping us to develop. I mean, these are some of the students and postdocs who worked on this program. The ones that are circled are still in my lab. I would like to acknowledge, of course, the funders of our research. Please note, if you don't have any research funding, you cannot do all the things I've shown to you. So research funding is very important, and I would say that, the Nigerian government should be aware that even in the United States and all the industrialized countries, almost all the important research funding for research in universities and higher learning, all the funding comes from government, not private sector. That's very important for you to know. Mr. Vice Chancellor. Yes. All right, so I'll end by just thanking all of you. Thank the Vice Chancellor. Thank the Dean. I thank everybody. Thank you.